So Telltale of COVID-19 apocalypse. It was 31st December 2019, the New Year Eve. China WHO office was informed of a pneumonia-like outbreak of unknown etiologies. A bunch of 40-odd people from Wuhan city complained of fever, respiratory distress, cough, fatigue, and they were exposed to Wuhan seafood market, in which live animals like bats and pangolins are traded. On 12 January, China shared the genomic sequence of this novel coronavirus as beta coronavirus. On 24 Jan, Lancet published its first paper stating the clinical manifestation of these patients. They had high cytokine chemokine expression, lymphopenia with a reduced CD4, CD8 T cell counts, high D dimer is a degradation product of fibrin, a readout of hypercoagulation, the hallmark of severe COVID patients. The patients also had groundless opacities in chest CT, accounting for the ARDS. They were also comorbid for diabetes, hypertension, cardiovascular diseases. On 29 Jan, another paper came out which states the similarity of this virus with the bat-derived virus and is phylogenetically distant from SARS and MERS. These bats are actually asymptomatically known to harbor many RNA zoonotic viruses as Ebola, Nipah, rabies, SARS, and MERS. What is so special about these bats? The bats overall has a dampened inflammatory response. The Pyhin genes are absent from the genome, accounting for the absence of DNA sensor like AIM2, IFI6. The STING pathway is also downregulated by the mutation of the conserved S358 residue. The TNF alpha is also downregulated by the presence of C-rail repressor motif in the promoter, and the NLRP3 also downregulated by impaired transcriptional priming. So overall, the pathways are downregulated. Only interferon alpha is constitutively active. So that perhaps accounts for taking care of the virus while harboring. Now, in early Jan, this virus was restricted only to China. By the end of the March, it spread all throughout the world, with a pandemic declaration by WHO on 11th March. Why this is so infectious? The spike protein of the CoV2 binds 10 to 20 times stronger than the spike protein of CoV to AC2 receptor, and the presence of furin recognition motif in CoV2 is also accounting for its so high infectious nature. The course of the infection, primarily 80% of the patients who are RT-PCR positive, they are asymptomatic, with the remaining 20% in stage 2 and stage 3, they become symptomatic and they perhaps need hospital intervention. So pathophysiology. For the mild and moderate, the type 1 interferon is fairly in the higher limit, so which actually takes care of the infection. The lymphocyte is in normal range. The inflammatory cytokines are not at all activated, and the viral load is also not much in the higher limit. But as the disease progresses to a severity, the viral load picks up. The type 1 interferon response goes down, which actually is responsible for exacerbating the pathology. And the inflammatory cytokine ramps up, which causes the major damage. And the lymphocyte goes down, which actually attributes for the chances of secondary infection. So overall, the immune system gets deregulated. Now, the type 1 interferon and the IHCs are known to be suppressed in severe and critical COVID patients. So as we see here, when we move from severe to critical, the interferon activity goes down, and also for the IHCs. So perhaps the virus is outsmarting the host antiviral system of interferon and IHCs by suppressing it. And this is a similar strategy which is being resorted to or employed by influenza and the dengue virus. So they do it by the expression of NSP1. So NSP1, when transfected into HEC2 T cells, they inhibit the translation of luciferase, but the mutant one, they do not repress in response to a challenge with the Sendai virus. So as we move from moderate to severe, there is a hyperstimulation of the cytokines, as we see here from IL-1, IL-6, TNF-alpha, CXC, CCL, all these chemokines which are responsible for exacerbating the lung pathology. Interesting results came out, which says that the more number of days the patient stay in the hospital, the more amount of cytokine expression is there. So it is reflected in interferon alpha-2, TNF-alpha, IL-17, IL-1 receptor antagonist, IL-1 beta, IL-6. And it also shows that the deceased patient or the patients who died have more amount of cytokine expression than the people who are discharged from the hospitals. So interestingly, interferon alpha is expressed here. So perhaps interferon alpha in this result is unable to control the viral load. It also shows that interferon lambda to be present in severe patients, which I will talk later. Interferon lambda plays a crucial role in actually disrupting the epithelial barrier junctions. Also, interestingly, interferon IL-1 receptor antagonist has been shown to be upregulated in severe COVID patients. So in the range of 1000 picogram per ml. So that's a fairly high amount. So IL-1 receptor antagonist is a natural blocker of IL-1 signaling. So it actually gets upregulated during inflammations to put a brake on the IL-1 signaling. So perhaps IL-1 signaling plays an important role in COVID pathogenesis. So the characteristic pathogenesis of severe and critical COVID patients: compromised epithelial endothelial barrier, cytokine storm, the aftermath of the deregulated immune system, pleural effusion due to accumulation of hyaluronic acid, and fibrosis due to deposition of collagen, vascular endothelitis. The endothelial cells get damaged, and a respiratory virus hitting the endothelial cells is not much known of. This is an exception. Hypercoagulability leading to DIC, thrombosis, and ARDS-like manifestations, and eventually sepsis and multi-organ failure. So it's interferon lambda which is actually disrupting the lung epithelial barrier. When poly-IC-TLR3 agonist is actually instilled into the trachea, there is more amount of protein in the bulb fluid, but RH48, the TLR7, doesn't do this. And in fact, the IFNAR interferon lambda receptor knockout seems to be protected in comparison to the wild type. And this lung epithelial barrier disruption actually increases the propensity for secondary infections. And in COVID, actually, we know that secondary bacterial and fungal infections are exacerbating the whole pathophysiological condition. The dead cells actually initiate inflammation in an IL-1 dependent way. So when the neutralizing antibodies to anti-IL-1 alpha was injected, the neutrophil influx was reduced in comparison to the IL-1 beta. And also the IL-1 receptor knockout showed much decreased neutrophil influx. IL-1 alpha also plays a critical role in the blood coagulation. When the P33, the pro-IL-1 alpha, was actually incubated with thrombin, and it was actually done in HeLa cells, you see a good amount of production in IL-6, but when it was incubated in presence of a neutralizing antibody, the IL-6 production goes down. And also the active IL-1, the P18, was found to be present in septic patients with ARDS. So this presence of P18, or the active form IL-1, plays an important role in the biological phenomena. So what happens in a necrotic cell, when the IL-1 alpha is actually liberated, it actually binds to IL-1 receptor and thereafter transmits a downstream signaling to produce CXCL chemokines and IL-6, which actually promote the attraction of macrophages and monocytes into the lungs and further exacerbating the lung pathophysiology. So it can also work in a paracrine fashion by inducing, by binding to IL-1 receptor and then promoting IL-1 beta and actually amplifying the whole inflammation loop. Therapeutic interventions. So I would perhaps talk about the only repurposing, because as of now, the vaccines are not yet ready. For the asymptomatic, perhaps only antivirals, interferon beta and remdesivir would be perhaps good, because interferon has shown some good results, and interferon, if provided early, can be really efficacious, because these viruses actually, they dampen the interferon response. For severe and critical, perhaps a communal approach of anti-inflammatory to tame the cytokine storm along with antiviral and anticoagulant as heparin, depending upon the severity. So this COVID has actually similarities with autoimmune diseases. So autoimmune diseases and anakinra, an autoimmune drug, which is actually used for rheumatoid arthritis, has shown much promise in smaller clinical studies. And anakinra as anakinra blocks both IL-1 alpha and IL-1 beta, so it can be really a good molecule for testing in COVID cases. And Blocking gas turbines and gas spaces can be a good target too, which works upstream in the inflammasome pathway. And some other cytokine blockers like GM-CSF, TGF, R6 can be good too. So to wind up, the suppression of the interferons coupled with the hyperstimulation of cytokines lead to severity of COVID, and R1 can be the master regulator of the inflammatory cascade, though more studies are required. And about 23 clinical trials are being conducted now by NIH to see the effect. I greatly acknowledge the initial insightful discussions with Dr. Fitzgerald and Dr. Goldenbaum. I'm thankful to Dr. Tomar for the video edit.